plant biotechnology represents a powerful suite of techniques designed for the genetic modification and improvement of plants. It encompasses a broad range of scientific approaches that manipulate plant biology at the molecular level. This field is crucial for advancing agricultural practices and addressing global challenges. The core purpose of plant biotechnology is to offer innovative solutions to critical global issues. These include enhancing agricultural productivity, ensuring food security for a growing population, and promoting environmental sustainability. By leveraging biological processes, we can develop more resilient and efficient plant systems. The primary goal of these biotechnological interventions is to introduce desirable traits into crops. This can lead to significantly increased yields, allowing more food to be produced from the same amount of land. It also aims to confer pest resistance, reducing crop losses and the need for chemical interventions. Furthermore, plant biotechnology seeks to enhance the nutritional value of food, combating deficiencies worldwide. One of the cornerstone techniques in plant biotechnology is recombinant DNA technology. This process involves the isolation of specific genes and their subsequent insertion into a plant's genome. It allows for the precise transfer of genetic information across different organisms. The key steps in recombinant DNA technology begin with the isolation of a gene of interest. This gene can originate from virtually any source, whether it be another plant, an animal, or even a microbe. Once isolated, the gene is then inserted into a suitable vector, such as a plasmid or a virus, which acts as a carrier. This modified vector is then introduced into plant cells, facilitating the delivery of the new genetic material. The ultimate goal is the integration of this foreign gene into the plant's own genome. If successful, the plant will then express the desired trait, leading to a new or enhanced characteristic. Common applications of recombinant DNA technology have revolutionized agriculture. It has enabled the creation of herbicide-resistant crops, simplifying weed management for farmers. This technology is also behind the development of insect-resistant crops, significantly reducing damage from pests. Moreover, it has been used to engineer virus-resistant crops, protecting them from devastating plant diseases. Another fundamental technique is tissue culture, which involves the propagation of plants from small pieces of tissue. This process is carried out in vitro, meaning in a controlled laboratory environment, often on a nutrient medium. Tissue culture allows for precise control over plant growth and development. The primary uses of tissue culture are manifold. It enables the rapid multiplication of plants that possess highly desirable traits, essentially cloning them to produce genetically identical copies. This technique is also invaluable for the production of disease-free plants, ensuring healthy starting material for cultivation. A related technique is protoplast fusion, which involves the fusion of plant cells that have had their cell walls removed. These naked plant cells, known as protoplasts, can then be induced to merge. The objective of protoplast fusion is to combine genetic material from different plant species. This method offers a significant advantage by enabling genetic combinations between species that cannot be crossed naturally through conventional breeding. It bypasses sexual reproduction barriers, opening up new possibilities for creating novel plant varieties. Protoplast fusion expands the genetic diversity available for crop improvement. Genetic transformation methods are crucial for introducing foreign genes into plants. One of the most widely used techniques is agrobacterium-mediated transformation. This method utilizes Agrobacterium tumefaciens, a naturally occurring soil bacterium. The mechanism relies on Agrobacterium's natural ability to transfer a piece of its own DNA, known as tDNA, into plant cells. In a remarkable feat of scientific exploitation, researchers replaced the natural tDNA with the gene of interest they wished to introduce. This engineered bacterium then acts as a biological delivery system. This method provides an efficient and precise way for gene transfer into a wide range of plant species. Another significant method is the gene gun, also known as biolistics. This technique involves microscopic gold or tungsten particles that are coated with the DNA containing the gene of interest. These DNA-coated particles are then physically shot into plant cells at high velocity. The gene gun is particularly useful for transforming plants that are not susceptible to agrobacterium-mediated transformation. It offers a direct physical means of introducing genetic material, bypassing biological barriers. This versatility makes it an important tool in plant biotechnology research and development. The application of these biotechnological techniques has led to significant outcomes in agriculture, particularly through genetically modified, or GM, crops. These advancements have profoundly impacted food production and farming practices globally. One of the most significant outcomes is the improvement in crop yields. By engineering plants to be more robust or efficient, GM crops can produce more food per acre. This contributes directly to global food security and helps meet the demands of a growing population. Another crucial benefit is the reduced reliance on chemical pesticides. Many GM crops are engineered to be resistant to pests, meaning they can defend themselves. This leads to fewer chemical sprays, which is beneficial for both the environment and human health. Furthermore, GM crops have enabled the enhanced nutritional content of food crops. This addresses specific dietary deficiencies in various populations, improving public health outcomes. Illustrative examples highlight the impact of GM crops. Bt corn is a prime example, engineered to produce its own insecticide. This internal defense mechanism significantly reduces the need for external chemical sprays, offering a more sustainable pest management solution. Another groundbreaking example is golden rice, developed to produce beta-carotene. Beta-carotene is a precursor to vitamin A, and its presence in rice helps address vitamin A deficiency, a major public health issue in many developing countries. These examples demonstrate the potential of biotechnology to solve critical agricultural and health challenges. Despite the immense potential, plant biotechnology and GM crops face several challenges and considerations. These require careful evaluation and ongoing research to ensure responsible development and deployment. One area of concern involves potential environmental impacts. There is a risk of developing herbicide-resistant weeds if herbicide-tolerant crops are not managed properly. This could lead to a cycle of increasing herbicide use. Concerns also exist regarding the effects of GM crops on non-target organisms, such as beneficial insects. Understanding these interactions is vital for maintaining ecological balance. Therefore, careful consideration and rigorous testing are absolutely essential before new GM crops are released. Beyond environmental aspects, societal and regulatory hurdles play a significant role. Public perception and acceptance of GM crops vary widely across different regions and cultures. Misinformation and lack of understanding can hinder adoption. Complex regulatory frameworks govern the development and deployment of GM crops in many countries. These regulations are designed to ensure safety but can also create lengthy and costly approval processes. These factors significantly influence the adoption and acceptance of GM crops, shaping their global impact. In conclusion, plant biotechnology remains a powerful and indispensable tool in modern agriculture. It offers innovative solutions to some of the most pressing global challenges of our time. By harnessing the power of genetic modification, we can continue to improve crop characteristics and resilience. This field is crucial for addressing global food security, ensuring that enough nutritious food is available for everyone. It also plays a vital role in promoting sustainable agriculture, reducing environmental impact, and fostering more efficient farming practices. The ongoing advancements in plant biotechnology hold immense promise for a healthier and more sustainable future.